Alright guys, this is chapter 6. We're going to be talking about lipids. We're going to be talking about triglycerides, phospholipids, and steroids. We're mostly going to be talking about these phospholipids because they make up cell membranes. Alright, and that's what the focus of this chapter is on cell membranes. So I'm going to cover sections 1 through 4. 4 is going to be covered vaguely. What I'm going to want you to do is actually read chapter 4, create your own notes for chapter 4, and then present them to me. That'll be your assignment. So I'm going to cover chapter 4 very vaguely about transport proteins and so forth. But in this lecture, I'm going to cover sections 6.1 through 6.3, which covers lipid structure and function, the lipid bilayers, and how stuff moves across the lipid bilayers, which is your cell membrane. All right. Here we have lipids. Here's a triglyceride made up of fatty acids, and here is actually a lipid bilayer. And I'll explain these throughout the lecture. First, we'll start off with lipids. Lipids are made up of fatty acids. Fatty acids are their monomers. Just like nucleic acids have nucleotides, proteins have amino acids as their monomer, carbohydrates have monosaccharides as their monomer. For lipids, their monomer is fatty acids. So these fatty acids are either going to be saturated or unsaturated. You've probably heard of these when people talk about the fat in food. When it's saturated, that means it's saturated with hydrogen. When it's unsaturated, you have this double bond that leaves a gap right here, so it's not saturated with hydrogens. And in food, you'll always hear that word, that polyunsaturated fats. That means they have more than one of these double bonds. There's 3 types of lipids. You have triglycerides, phospholipids, and steroids. Triglycerides are actually made up of 3 fatty acids linked together by glycerol. A phospholipid, kind of the same thing, has 2 fatty acids linked by glycerol, but yet have a phosphate head. Okay. Now a steroid is completely different. Steroid, its basic structure is going to have 4 rings: 1, 2, 3, 4, and then at both ends you're going to have different types of functional groups. Okay. So we'll start off with the steroids. The most common steroid of all is cholesterol. Cholesterol, once again, has these 4 rings and then has a functional group here and throughout its structure, but its basic skeleton structure is 4 rings. Here you have estradiol, where estrogen is formed from. It's a derivative of estrogen. Then you have testosterone, which is also a hormone, and you have cortisol. All right. So once again, steroids are bulky. They have 4 ring structures. They have different functional groups attached to them, and here are the different examples. The other ones are fats. Fats are nonpolar molecules. They're made up of 3 fatty acids, which are triglycerides. Their primary function is to store energy. They're formed by dehydration reactions, and their linkage is called an ester linkage, the way they're linked together. So remember, glycerol, all right, you have your 3 molecules here, and then you have 3 monomers, which are fatty acids. They go through dehydration synthesis. You remove water, and then you'll link these groups together, and now you have a triglyceride, which are your fats. Then you have phospholipids. Phospholipids are what make up the cell membrane. A cell would not be a cell without the cell membrane. So cell membranes are made up of phospholipids. They're composed of glycerol linked to a phosphate group and then to a hydrocarbon, which are the fatty acid tails. All right. So here you have your polar group, which is your phosphate. All right. This part of the fatty acid loves water. Okay. This part of the phospholipid loves water. The fatty acid tails, as you know, fat is not soluble in water, does not dissolve in water, so it hates water. All right, so it's hydrophobic. All right, it's hydrophobic. So here you have your lipid bilayer. Okay, your phospholipid bilayer. You have your hydrophobic tail, which is made up of these fatty acids, and you have your hydrophilic head, which is made up of the phosphate group. Okay. Extracellular means on the outside of the cell. Intracellular means on the inside of the cell. All right, here in this video, you're going to see what happens when phospholipids are placed in a solution, usually a solution of water. Since they don't like water, they're not going to face the water side. They're going to form this bilayer, and you'll see this really cool model here. So the fatty acid chains are the green, and the polar heads are over here with the gray, the blue, and so forth. And you can see them start clumping together, trying to get away from that water, these fatty acid tails. But yet they're still attracted to each other. And now you can see the lipid bilayer starting to form. And this is an actual model they've done studies on this, and this actually happens when phospholipids are placed in water. They form these bilayers and forming a sphere, which ends up making the outside part of a cell. So there you have it, your phospholipid bilayer. Your fatty acid tails, which are hydrophobic, and then you have your phosphate heads, which are hydrophilic. And all these up here are molecules of water. As you can see, water is not found within the bilayer. It is kept out, but yet it can still move. It could still move across if needed, which we'll explain later. All right, the cell membrane has other names in case you hear it. It has a fluid mosaic model. It has a plasma membrane and a lipid bilayer. Okay. The main function of a cell membrane is to maintain homeostasis within the cell, and that's going to happen by being selectively permeable. So characteristics of the cell membrane: it surrounds the cell, made up of phospholipids, made up of two layers, also known as a bilayer. It's selectively permeable. What that means, it controls or selects what can enter or leave the cell based on its chemical composition. Some materials are allowed in, some others are not. All right, its main job is to maintain balance within the cell, internal balance within the cell, balance and equilibrium. Selective permeability, okay? So there's certain things that can move across the cell membrane without any problem. There's certain things that cannot, and that's what this little illustration shows. So small nonpolar molecules like oxygen, carbon dioxide, and nitrogen are able to move across the cell membrane without any problem. All right, so they're highly permeable, okay? Or they have high permeability. Then here you have small uncharged polar molecules, okay, like water and glycerol. Those are also able to move across without any problem. But then you get the larger molecules, and then uncharged polar molecules such as glucose and sucrose. These guys can't move across easily, all right? So they have a low level of permeability to the cell membrane. And then ions such as the chlorine ion, the chloride ion, and the potassium ion, and the sodium ion, these cannot move across the cell membrane. They need help. So we're going to talk about those in the next few, in a couple of slides more. So remember, it's selectively permeable, so certain things can move across while certain things can't. All right, and the cell membrane controls all this through its lipid bilayer. The fluid mosaic model, that means it's made up of different molecules, just as a piece of art called a mosaic, made up of all these different little pieces, which make up a bigger piece. That's the same way this the cell membrane works. It's made up of phospholipids, proteins, cholesterol, and the one I forgot here is also carbohydrates. Right? These molecules aren't static; they're not still, but are fluid, so they're able to move around, and you'll get to see this in this quick video.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins, which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
All right, that was a quick little video of the fluid mosaic model, or the cell membrane, plasma membrane, or the lipid bilayer. All right, just remember its components. It's made up of fatty acids. A phospholipid is made up of fatty acids and a polar head. The polar head's hydrophilic, meaning that it actually likes water. The hydrophobic tail, once again, as you know, fats do not dissolve in water, so they don't like water. So they're hydrophobic. They form these lipid bilayers, as you saw the video earlier. just know that cholesterol is found within the cell membrane. What cholesterol does, it helps and it adds extra support to the cell membrane. So remember, these are still considered lipids. Remember the 4-ring structure, its basic structure, cholesterol. So we do need cholesterol in our bodies, but yet too much will harm us. So we need cholesterol for hormones, and we also need cholesterol for the stability of our cell membranes. Just know also that in this fluid mosaic model that there's also transport proteins. These proteins actually allow help stuff to move in and out of cells. As you can see here, there are transmembrane proteins. That means they go across the membrane. This is a receptor protein. This is a channel protein. This is a gated channel protein because it has a gate right here. And this is just another transport protein. And then you have your glycoprotein, which means you have a carbohydrate like you saw in the previous chapter, a carbohydrate, a functional group attached to a protein, and this is used for signaling. And then you can see the cholesterol right in here, offering that stability within the cell membrane. Once again, signal proteins, they assist the cell in communicating with the outside environment. So you have a glycoprotein. So you have a protein with a carbohydrate chain on top of it. And then you don't forget about the glycolipids. Glycolipids, instead of being attached to a protein, is attached to a phospholipid. So it's called a glycolipid. So in this next section, we're going to talk about how stuff moves across the bilayer. So I'm going to cover a little bit of section 4 here, but once again, I want you to read section 4 and create your notes, and you're going to present them to me. But I'm going to briefly go over some of the stuff from chapter 4, but not into too much detail like they do in the reading. So I want you to read, break down chapter 4, and create your notes, but use this lecture as a foundation for it. Okay? So there are 2 types of transport. You have passive transport, you have active transport. Passive transport, you're actually moving molecules from high concentration to low concentration. Main one is diffusion. Okay? Diffusion, you have molecules of high concentration here, and they move across the cell membrane to low concentration. That means they're going with a concentration gradient, right? Without energy. That's what it means to be passive, without energy. Or you can have help from these proteins like we mentioned before. These proteins actually still move molecules from high concentration to low, all right? From high concentration to low, but these proteins do not need energy to move these molecules across the cell membrane. They're just there, what they call facilitated diffusion. They're just there to help. All right, just there to help. That's what facilitate means, to help, to offer some support. So what they do, they once again in passive transport, in all 3 of them right here, all 3 examples that you see here, you're moving molecules from high concentration to low concentration with the concentration gradient. It's like going down a hill. All right, then you have active transport. Active transport actually moves molecules from low concentration to high concentration with energy. Remember, that's our energy coin, ATP. So passive transport, this is a perfect thing of passive transport. If you ever thrown yourself down a hill, you really don't need much energy. You just go down and that is it. So the movement of molecules from high concentration to low across the cell membrane. They go down their concentration gradient, no energy required. All right. Diffusion, as you can see, these molecules are diffusing back and forth across the cell membrane. So you go from high concentration to low. It's a form of passive transport. And it's moving with the concentration gradient, and it's mostly gases that do this. All right. Then you have facilitated diffusion. Here you have an example of facilitated diffusion where no energy is required, and it's moving molecules from high concentration to low concentration. Okay. So now it's balanced. You have 4 molecules on each side, so it's at equilibrium. That's what the cell membrane is trying to establish. That's what its main job is, to maintain that homeostasis. Here you have again these transport proteins. No energy required, going from high concentration to low concentration. That is with the concentration gradient. Now osmosis. Osmosis is just like diffusion, but you're moving water. All right. Anytime you hear the word osmosis, the movement of water from high concentration to low concentration with the concentration gradient. Okay. No energy required as well. It's a form of passive transport. Then this produces osmotic pressure. It's pressure the cell undergoes depending on the type of solution they are in. There are 3 types of solution. You have hypertonic, isotonic, and hypotonic solutions. Okay. Here's a great illustration of what happens when a cell is placed in these different solutions. If placed in a hypertonic solution, it will shrivel. If placed in a hypotonic solution, it will burst. If it's at homeostasis, then it's in an isotonic solution, as you can see here. Hypertonic, isotonic, hypotonic. So let's go over each one of them. In a hypertonic solution means that there's more solutes or salt outside the cell. And the rule you have to remember for this is that salt sucks. All right, salt sucks water. So the amount of solutes or salt is greater outside the cell. Solutes suck water out of the cell shrivels and shrinks. So it's leaving the cell. And look at the difference between an animal cell and a plant cell. A plant cell still has a cell wall around it. So the cell shrinks. Nothing really happens to the cell wall, but the cell shrinks. But yet, it doesn't have the pressure to hold up the cell wall. Okay? So that's in a hypertonic solution. Once again, more salt or solutes outside the cell. Salts and solutes suck water, so it sucks water out of the cell, so they shrink and shrivel. In an isotonic solution, you have homeostasis. The amount of water and solutes are at equilibrium. It's at homeostasis. The same amount of water coming in is the same amount of water going out. As you can see in these two types of cells here, your animal cell and your plant cell. In a hypotonic solution, there's more solutes inside the cell than outside the cell. Since solutes suck water, salt sucks water, then water will rush into the cell. The cell will swell up and lyse. That means it will burst. That's the whole thing between hypotonic. It swells up like a hippo. Hypo-hippo. Alright, so now let's take a look at this question. Why does eating popcorn make you thirsty? I want you to think about it for a few seconds. Alright, you go to the movies, you have this big tub of popcorn, all of a sudden you're thirsty, and why? Here's the answer. Popcorn is very salty. Salt sucks water and may cause the water to leave the cells in your mouth due to diffusion, and this makes a person thirsty. And what the crazy part is that you drink soda, and now soda has more salt, so guess what you start doing is just a cycle. You start drinking more and more soda, so you want to go buy more, so they sell more. So think about it next time you go to the movies. Alright, now we're going to go into active transport. Active transport is the movement of molecules from low concentration to high concentration using ATP, which is energy. You're going against or up the concentration gradient as opposed to passive transport. Passive transport goes with the concentration gradient from high concentration to low. Now you're moving against the current. Just think about if you're in a river or a stream. And when you go along with a stream, you're floating down the stream. You're going with a concentration gradient. Now if you want to go back upstream, you have to use energy. You're pushing yourself, right? So you're using up a lot of energy. So it's active. This is active transport. Examples of active transport are molecular transports. The use of channel proteins with energy to get molecules in and out of the cell, also known as molecular pumps. So here's low concentration. So you want to move it. Diffusion usually happens from high to low. So now you want to move the stuff against the concentration gradient from low to high, but you need energy to move these molecules across. All right. I'm going to go back so you can see this little illustration again. All right. You're moving stuff across the membrane with energy. That's active transport. And a good example is the sodium-potassium pumps. Okay, sodium-potassium pumps. Once again, you're going to read chapter 4 or section 4 on your own, and you're going to create your own notes because they talk about these membrane proteins and how they're able to move these molecules across with energy. All right. Another form of active transport is endocytosis. The cells move substances or larger molecules into the cell, known as phagocytosis or pinocytosis. And this illustration is exactly what happens. The cell membrane pinches off, entrapping some of these molecules and take them in. These cell membranes dissolve or open up and release the molecules that the cell needs. So that's endocytosis. Now the opposite happens with exocytosis. Exocytosis is when you're actually pushing these little vesicles, all right? They're called vesicles. These vesicles out so you could send the molecules out. All right, that usually happens with a Golgi apparatus. That Golgi apparatus actually packages stuff up, and then they're pushed out of the cell through exocytosis. So both of these endocytosis, where when some larger substances come in, or molecules come in, and exocytosis, when actually larger molecules or these large package of molecules move out, both of these are forms of active transport. Okay, so that's the end of the chapter. So like I said, I've covered sections 1, 2, and 3, and 4 vaguely. So your job now is to go ahead and read the chapter, finish the reading quizzes, and then I want to see your notes for chapter 4 after the reading. I want to see your note-taking abilities while you read. All right, so I wish you guys the best of luck. Hope you enjoyed this video, and I'll see you guys in class.